Okay, good afternoon, everybody. It's 2 minutes after 12 o'clock at noon, so we are ready to start this seminar. We have more than 85 people, and more people are coming. Hopefully, we won't crash the site with all the numbers of attendees, and we'll be able to get through this. Thank you for attending this seminar. The seminar will be given by myself. I'm a professor of virology at the Department of Pathobiological Sciences here at the LSU School of Veterinary Medicine. The seminar is being broadcasted as part of our graduate seminar series and also throughout the Louisiana Biomedical Research Network, for which I am the principal investigator. The seminar will be given principally by myself, as well by our collaborator, Ilya Brodsky, who is the CEO of Pine Biotech, a company which is a spin-off of the University of Haifa Tober Bioinformatics Research Center. There are a couple different aspects of the work that require bioinformatic analysis, and we are involved with Ilya and the Tober in teaching right now a graduate level in transcriptomics, RNA-seq work, as well as putting together a introductory course to bioinformatics. The name of that course is called Bytes and Molecules, which already has been approved in collaboration with Dr. Brilinski for the Department of Biological Sciences for LSU undergraduates. And we hope that this course also will be disseminated through the LBRN network and mirrored in all our member institutions throughout the state of Louisiana. So what I'd like to do today is go over some of the very basic molecular and cell biology of coronaviruses, visiting the importance of this epidemic. And also I added certain aspects of immunopathogenesis of coronaviruses that are quite important as we move forward to try to develop vaccines against the viruses. As part of our presentation, we will disclose some new findings from our laboratory in collaboration with one of the LBRN campuses and Dr. Seetharama Jois of the University of Louisiana Monroe, and that will happen towards the end of this presentation. So let me start then by really stressing the importance of infections throughout the world. And if you look at the top causes of death worldwide, with more than 56 million people that died in 2016, infections, direct or indirect effects of infections is by far the most important cause of death worldwide. Of course, circulatory systems with cardiovascular diseases and cancer being also very important. And if you look at the top 10 causes of deaths in low-income countries, and I think that will be important as you see later in a couple slides further down, you can see that low respiratory infections and infectious disease in general are by far the most important in terms of causes of deaths in low-income countries. And if you compare those to lower middle income, you'll see that the bars now shift, and ischemic heart disease is much more important, although low respiratory infections becomes less important. And if you look at the more upper middle income countries, you can see the respiratory infections being even of less importance than it would be for this particular incomes, the countries, whether they're really poor or not. And that really is quite pertinent because I listened to a seminar a few years back that indicated what is the big issue about disparities in health. And obviously the mother of all evils is actually poverty. And poverty is really the very basic societal description that ultimately relates to poor health. And this is particularly evident in this article that appeared recently by Rishana Ray on why are blacks dying at higher rates from COVID-19, this new pandemic. And in Louisiana, what we have noted that blacks represent about 1/3 of the state population, but really more than 70% of COVID-19 deaths. Most of these deaths have been centered in the New Orleans area, and there are specific probably reasons why it is, including the proximity of people, Mardi Gras festivals, and so on. In Milwaukee, the same thing. The Milwaukee County, Wisconsin, blacks represent 45% of that diagnosis, but over 70% of the deaths are related to COVID-19. And the author argues in this article that structural conditions that inform pre-existing conditions and health disparities are really the main culprit for the epidemic within the pandemic, which is ravaging Black communities across the US. So visiting now emerging and re-emerging diseases, we know over the years that we have a number of important diseases that came either from mosquitoes or through insects, a number of different animals, including from Rift Valley fever, 1931, Zika appearing first in 1947, and ultimately Ebola through bats, and Nipah viruses also through bats. And then more recently, 2003, the SARS coronavirus that had an intermediate host of the Himalayan civets and ultimately originated from bats. And of course, the bat origin of these newer viruses is not particularly surprising because there are more than 600 different genotypes of coronaviruses in bats. So transmission from bats to intermediate host, amplification, and then transmission to human beings could be quite possible because of selection of a particular variant that could be able to infect humans. And again, in 2012, we had MERS that also originated from bats, but it came through the documentary camels, and it was mostly concentrated in South Arabia. And of course, the prediction was that we'll have additional emerging diseases. The disease X would appear, and that was probably delayed. And now it's not really a surprise that we have the SARS-2 or SARS CoV coronavirus 2, which is related to the MERS and SARS virus, but it's quite different in terms of genotypic and phenotypic, as well as disease causing properties. So these are really the three major viruses in the 21st century. The severe acute respiratory syndrome virus, or SARS, it appeared in Guangdong province of China. It belongs to the genera beta coronavirus. It was transmitted again from bats to civets to humans. And it started in 2003 with an outbreak lasting until July 2003. There were more than 8,000 total cases, 774 deaths, and a fatality rate of approximately 9.6%, fairly high. The Middle East respiratory syndrome, or MERS, appeared in 2012, mostly in Saudi Arabia. About 80% of the cases were in Saudi Arabia. It's also in the genera beta coronavirus, was transmitted from camels to humans. There were more than 2,400 cases, 858 deaths, with a fatality rate of 34.4%. There are only a few cases here in the States of MERS, and those really from hospital people that were traveled from Saudi Arabia to the US. Now we have the 2019, the novel coronavirus, the coronavirus 2 that caused the coronavirus disease, or COVID-19, from the year that appeared. It appeared in Wuhan, Hubei province of China. It's the 7th human coronavirus caused illness in humans, and it's a novel beta coronavirus. There are 4 other human coronaviruses that I will visit later that also cause respiratory disease. Approximately 10 to 15% of all respiratory disease in humans, it's caused by benign human coronaviruses that are really cousins of the coronavirus 19 that we see here. So this is what appeared in 2003 in Time magazine, and it was titled, "How Scared Should You Be? The Battle to Beat the Deadly Pneumonia Virus." So really, in so many ways, we were warned in 2003 and warned again in 2012 and 13 that these coronaviruses could emerge and cause substantial disease, and yet the world seemed to be totally unprepared in facing this pandemic. And there are a number of reasons for that, including socio-political and others, including potential funding. I can tell you back in 2005, we submitted a project grant to NIH to look at antivirals against SARS. And of course, by then, SARS was not important anymore and the funding dried up. And again, it would have been very important to continue working on this type of viruses in the longer run. So this is the civet in markets, open markets in China. That's where SARS ultimately became the intermediate host, and it was amplified and transmitted to human beings. And of course, the MERS had a similar intermediate host with the camels, amplified there, and that's why the proximity of infections in Saudi Arabia. The more important aspect of SARS and MERS was that it required really close contact and potentially a high load, a viral load, to really establish those infections, which apparently it's not now true for the new coronavirus. It can be transmitted much more efficiently, and there are molecular reasons that we visit why this coronavirus maybe it is much more infectious and much more transmittable than the SARS and MERS coronavirus. This is a X-ray of a SARS patient back in 2003 that a 76-year-old man was exposed in March 7, ultimately died in March 21st within a few weeks of the infection. High temperatures, it showed the right upper lobe and bivascular interstitial infiltrates, and then a non-productive cough, dyspnea, and ultimately he was admitted to hospital March 16. It demonstrated the bilateral patchy air space disease, and then ultimately he was intubated and received mechanical ventilation, and progressive failure, respiratory failure, caused the death on March 21st. This particular pneumonia is reminiscent of what we see now with COVID-19, the SARS-2 coronavirus, but there are substantial differences, and the pathologists that are listening in probably can identify those, because the new pneumonias seem to be very different on X-ray images, starting at the lower part of the lung and then expanding outwards, having some sort of a glassy appearance that is quite different than the original SARS infection. So it does show that the new coronavirus, the SARS-2 coronavirus, causes a different type of disease pattern, and perhaps that's why it's much more acute in becoming much more lethal. So this is some histopathology slides from Fenner's virology book that shows the upper two panels shows SARS, and the top two, the bottom panels, are really from MERS. And you can see the lesions that have diffuse alveolar damage and progress to severity. In terms of severity, you see interstitial mononuclear inflammatory infiltration, and ultimately you see the on the right upper panel in the histo against the SARS proteins, you can see the formation of small syncytia, that is cells, multinucleated cells. The virus is able to really fuse cells together to create these multinucleated cells that are called syncytia. And this is the hallmark of these infections. And you'll see later how that differs, at least in cell culture, between the old SARS virus and the new SARS-2 coronavirus. And the acute disease, it's also noted in MERS, as you see here in the slides. So if we visit the numbers, I was looking at the numbers while I was preparing this presentation, and that on April 15, we had over 2 million of infections of COVID-19. The number of deaths was 126, over 106,000, and a mean infection rate of 2.5. It was so-called R0, or 2.5, meaning one person could infect up to 2.5 people. And then last night, that's probably within 24 plus hours, we see now that there's 2,060,000, 60,000 practically one day, with deaths now at 134,000. overnight. And in Louisiana, you can see that we have 21,000 confirmed cases and now over 1,000 deaths as of last night. And in the US, unfortunately, we have a high rate of infection, over 600,000 infected, with deaths now approaching the 30,000 mark, which is the highest in every country in the world at this point. And the recovery rate shown here worldwide, this is the positive note that the number of people have recovered, and at least presumably they're protected against subsequent infection with the same virus. And if you look at the characteristics of people that have been infected, at the low bottom on the red were deaths. You can see that the median age of death is 80 years old, with a span of 24 years to 105. And there is a slight ratio of higher for males, 58% in terms of deaths. And at least one underlying condition, 91% of the people had at least one underlying condition. And that's one of the hallmarks of really those infections. It takes advantage of any other comorbidity to really cause death. In terms of infections in general, the age in years is 54, much younger than the death, and with having age of 1 year old up to 105 also infected. And a lot of younger people have been infected and have had a tremendous disease issues, but most of them ultimately recovered. Again, you see a 52% male ratio versus female, and the healthcare workers, which is really a worrisome statistic, 13% of health workers, presumably having adequate protection, have also been infected. And this is part of the major issue in terms of the treating this disease, is really to protect the health workers, because ultimately these are the people that we depend on to overcome this disease. But I also want to tell you that, as probably most of you here at the vet school health school know, that coronaviruses for us, being in veterinary medicine, is not really particularly surprising, because there are a number of animal coronaviruses that cause substantial disease in just about every animal that you can encounter. And of course, there are coronaviruses in all vertebrates and invertebrates. They have a very wide span as a family. So in the original groups 1 and 2 and 3, which really represent the genera alpha, beta, and gamma, you can see that you have porcine coronavirus, you have feline infectious peritonitis virus, and transmissible gastroenteritis virus up on top. And it's the same group with the human coronavirus 229E, who causes a benign upper respiratory disease. On the beta genera, you can see human coronavirus OC43, and this is really a virus which is very close to, genetically at least, by evolutionary comparisons, to bovine coronavirus, and it does cause also both respiratory and enteric disease. And this is one of the hallmarks of these viruses, as you see, that it causes both respiratory and enteric disease. And as we know from COVID-19, SARS-2 coronavirus, there are indications that the virus may cause diarrhea in a number of patients that are affected with this virus. And of course, there are multiple other organs that are infected. And I think one of the issues with the SARS virus, the SARS-2 coronavirus and SARS, is the fact that not only it affected lungs and maybe the enteric tissues, but also a number of other tissues, including kidneys. And a lot of patients actually have died from kidney failure. And right now, dialysis equipment for kidneys, kidney help is really in short supply. And everybody's trying to also, in addition to ventilators, secure these types of equipment. This again from Fenner's Virology. I'm not going to really elaborate on this, but I wanted to actually show you that the number of different coronavirinae belonging to the alpha corona, beta, and the gamma and delta coronaviruses really cause a number of diseases. If you look at the canine coronavirus, it has a mild gastroenteritis and diarrhea. It can be transmitted by fecal oral route. And it's also noted here where there are vaccines. Some of the vaccines, and I will visit it again for animals, have been successful in limiting those infections. But then again, everything that we see here, that the virus can cause enteric disease, it could potentially be transmitted through the fecal oral route. It's actually a hallmark of these coronaviruses, and it wouldn't be surprising that this is actually true for SARS-2 coronavirus. I also wanted to tell you that our laboratory in the 90s and early 2000s, we worked extensively on bovine respiratory infections, and in particular bovine coronavirus. And for cattle, one of the major economic diseases is really shipping fever, which is really acute respiratory disease and lung pneumonias in cattle. Those that are shipped in long distances, that develop this disease and actually die with great economic losses in the cattle industry. And the pathogens, the viral pathogens that are involved include bovine herpes virus 1, the paramyxoviruses, bovine parainfluenza, the morbilli, pneumovirus, bovine adenovirus, and bovine respiratory coronaviruses. And we worked on bovine respiratory coronaviruses for a number of years. We published over 7 publications in the 90s that are shown here. And we really, and I highlighted one publication, 2000, with Dr. Hans Storz, who was the department head at that point in my home department, that really went to great extents to make me believe that bovine coronaviruses were more important than bovine herpes virus in causing shipping fever. And in 2000, we published this paper that coronavirus and Pasteurella hemolytica infections in bovine shipping fever really are the causative agents fulfilling the Evans postulates as opposed to Cox postulates, because the experiment couldn't be done according to the Cox postulates. And part of the idea was that the seroprevalence, the isolation of this bovine coronavirus was much, much higher than any of the other viruses. In addition to that, in 2001, we published a paper on elucidation of the genomic nucleotide sequences of bovine and coronaviruses. And we refer to, as part of the advances in experimental medicine, medical biology, we actually refer to the ability of these viruses to really create quasi-species and very rapidly maybe mutate and accept mutations within the genome. And going through the replication of these viruses, you'll see that these coronaviruses do have the potential of actually causing substantial mutation rates that could be selected through the host. So that's the paper that I was just referred to. It was co-authored with Dr. Gorbalenia, who's a fairly very well-known molecular evolutionary biologist. So for coronaviruses then, as part of the introduction, to introduce you the virus, obviously, as you see, these viruses belong to the Nido or Nidovirales order. Nido means nest because of this, the nested messages that the virus creates. It's significant pathogens of both humans and animals. And the corona is really the prefix, comes from the Latin word of crown, and is named for the crown-like appearance, which is shown on the slides on the transmission electron micrographs that you see on the right. And those corona spikes are really the spike glycoprotein. It's actually a trimer that creates this globular structure protruding outside of the membrane of the virus. The classifications are in general alpha, beta, gamma, and delta, and the human coronaviruses are really alpha and beta coronaviruses. They are medium-sized viruses that envelope a positive strand RNA of approximately 30,000 bases, and it's one of the largest viral RNA that's known in terms of genome. And this is the early on taxonomy of nidoviruses with the coronaviridae, the Artidae, aeronoviridae, and as part of the coronaviruses, we have the virus that we've seen, including the infectious bronchitis virus, and that was the first virus that was really discovered, coronavirus, in 1926, and it caused widespread death among birds throughout the world. And this is the actual full taxonomy from the taxonomy group that appeared in Nature in 2020. And you see that coronaviruses, the real Riboviria, Nidovirales, Coronaviridae, Orthocoronaviria, Beta coronaviruses for those connoisseurs for taxonomy in comparison to the humans themselves. You could actually Google the nature for the taxonomy group, and you'll be able to see that very quickly. So this is, again, the description in terms of size: 30,000 bases of genome equivalent to the left, although some of the other viruses are much smaller, and they look very different, as you see the structure for torovirus, the arteroviridae, and the ronaviridae here. And this is, again, from a recent review article, the phylogeny of coronaviruses, that will show the different genera, alpha, beta, gamma, and delta. And you can see that the SARS coronavirus and the belongs to the beta-2 group, as including the SARS-2 coronavirus, and the MERS coronavirus is really on the beta-1 subgroup of the beta genera, and the other human coronaviruses as part of the alpha coronavirus, shown here at the yellow. And at this point, we have been working, again, as I mentioned earlier, in a collaboration with Pine Biotech and Eli Brodsky and the Tobin Bioinformatics Research Center. And we have access to the pipelines that Pine Biotech has developed to look and compare the molecular evolution of different viruses. And I invited Elia to share with us in trying to answer this very important question. Where did SARS-2 coronaviruses come from? So Elia, if you're listening, can you share your screen and take us from there?
Okay, thank you, Gus. So I think what is important to kind of look from a bioinformatics data perspective is that the speed at which data has been deposited and made available on NCBI and other platforms, genomic data of these different viruses, is accelerating really quickly. So you can see here, this is an average number of full human genomes, so human host genomes of different viruses, and COVID-19 is significantly higher. This is not so much talking about the total quantity of these genomes as to the speed that these genomes have been made publicly available. And so one of the interesting things for us collaborating with labs interested in these specific different viruses is to understand the diversity, the origins, and the other types of questions proposed by the research community. So what we did was develop a whole number of tools to be able to study the genomic data and try to integrate that data to really understand what's going on. So I will just briefly touch on a couple of different types of analyses that we've done to explore this question of origin, as well as mutations that have been accumulating over time as this epidemic continues to expand. So one of the tools that it's going to be interesting to look at here in the context of genomic data that is available. Genomic data could be as assembled genomes, so genomes that have already been identified as a consensus from one or multiple samples that have been collected, as well as the raw sequencing data that comes from patients. So I'll just give a couple of examples in the context of this presentation. One of the initial analyses that could be done to really understand the relationships between these genomic sequences is to take whole genomes and align them using multiple sequence alignment, and then understand phylogenetic relationship between, for example, previous outbreaks or different subtypes of these different viruses, for example, MERS, SARS, and other beta coronaviruses, and which, for example, is the current COVID-19 sequence most closely related to. So by looking at the different methods as well as the different combinations of genomes, what we can clearly see is that COVID-19 is the closest, more closely related to the SARS coronavirus. And I think looking deeper into the sequence alignment, we can really start asking questions about how are they related and where are they different. And so those types of questions are easy to ask. We have a lot of data that is publicly available, and we can really know the origin of those samples as well as the different hosts. So we've done multiple comparisons between different different data sets, and I want to show today some specific examples of how they are different based on this analysis, which is first of all a comparison of SARS and the SARS-2 and where those sequences are different. So what you can see here is the frequency of mutations. Specifically here, you can see over the whole genome, and you can start seeing these different areas that show kind of significant differences. And if we expand some of those differences, we can start looking at specific regions where a lot of variation could be observed. And so this is the nucleotide sequence. And of course, one of the important questions when we look at nucleotide sequence is to ask, well, how is this different at the amino acid level? So what we've done is I'm quickly going to demonstrate a couple of examples. This is a comparison between the a number of sequences from human hosts from SARS and SARS-2, and you can see how there's an accumulation of changes throughout the spike glycoprotein. What's interesting though is that if we look at this comparison between, for example, the closely related bat coronavirus rad-G13 genome and the human novel coronavirus COVID-19 genome, those changes are starting to accumulate in very specific regions of the spike glycoprotein, which as we will talk later about the significance of the spike glycoprotein, if we look at this glycoprotein when it is in its binding state with the ACE2 receptor, we can see how those same mutations that we observed there start mapping directly at the interaction at the receptor binding domain between the spike glycoprotein and the ACE2 receptor. And so what's important to understand about this model is that the process by which the spike glycoprotein opens up and binds to this ACE2 receptor is actually something that a lot of people are looking into, and it's not very clear how significant mutations on the outside and on the inside of this receptor binding domain, what role do they actually play in this whole process? But what is obvious here is that the rat G13 genome from the bat coronavirus and the human Wuhan, so that same genome from the first published sequence from this outbreak, have a significant difference, specifically in that receptor binding domain area, which of course has resulted in proposed intermediate hosts that have a closer alignment between those sequences, specifically in the spike glycoprotein. So, I think as new data is coming out from different hosts and from different genomic sequences, as well as from patient sequences, what we are trying to work on together with the Louisiana Biomedical Research Network is to make this data easily accessible for other people to be able to look through and really understand the relationships between these genomes. So what we started doing, and I'm just going to quickly, so I just wanted to mention and summarize this portion of the presentation that a lot of these comparisons, I think it's easy to quickly understand the relationships between the genomic sequences. So we've put together a collection of different data sets that represent these comparisons. Here, what you can see is accumulation of mutations in bins. And for example, here, this is mapped on the reference genome from this outbreak. And you can see how the gray represents the least different, the red represents the most different. You can see the annotation of these different genomes by type. So there's MERS, beta coronaviruses, COVID-19 and then the original SARS. And you can see how those are mapped on the different proteins on the reference genome. For example, if we look at the surface glycoprotein, you can appreciate the structure of this protein and the function of this protein, and then look at the differences between those different types of genomes, for example, by expanding the view and looking at the nucleotide sequence and the amino acid sequence, and how that translates to specific differences between those different genomes. So briefly, this is just to summarize some of the analysis that really demonstrates how some of the closer related genomes from bats nevertheless show important mutations in the binding region with the ACE2 receptor, and some intermediate hosts like the pangolin that has a genome overall that has more differences from the human COVID-19 genome, but in that receptor binding domain has less dissimilarity. So that has been proposed as the intermediate host. Briefly, maybe here I will stop, Gus.
Okay, so basically what we, Elia did with these molecular evolution things, it does really relate the fact that it's close to bat virus, is related to coronavirus, but really at this point, we really do not know where it came directly from bad coronaviruses or through some kind of an intermediate host, or even really through human beings, because there is a possibility that anyway, so we don't really know whether it actually circulated in human beings and ultimately evolved much earlier and not causing disease. So this is the coronavirus, as we saw earlier. We have a lipid enveloped virus. It has the spike glycoproteins. This actually shows the bovine coronavirus. They have the hemagglutinin in addition to the E glycoprotein and the spike glycoprotein. There's a 30,000 nucleotide RNA positive strand genome, and it's covered by the N protein or the nucleocapsid protein. And we have additional the M, the matrix protein, and the E glycoprotein that are part of the envelope. And I'll be talking about those more specifically as we go on. This is the electron transmission electron micrograph of the virus itself from 1967, showing how the spikes can be easily seen, about 100 to 120 nanometers in diameter for the size of the virus. Again, plus-stranded RNA viruses, these are the groups that I already visited in, earlier, with the coronavirus having these animal viruses as part of the group. The relative number of structural proteins, for instance, bioparticle, you have the spike glycoprotein with over approximately 180 to 220 trimers, the membrane protein, the nucleocapsid protein, the small envelope protein being far less represented. This is a depiction of the life cycle of the virus. The virus enters by binding to specific receptors, and different genera use different receptors. We're going to see that aminopeptidase N, the ACE2, angiotensin converting enzyme 2 receptor, and the dipeptidopeptidase 4, all those are really receptors for different coronavirus. The virus can enter by fusion of the viral envelope with the cellular membrane, depositing directly the nucleocapsid into the cytoplasm. It can also enter, as is shown here, through endocytosis, and then in the endosomes, because of low pH, there is activation and cleavage of the spike protein, and the virus then fuses its viral envelope with the endosomal membrane to produce the nucleocapsid inside the cytoplasm, where the replication will occur. I'll be talking more specifically about the genome replication. Again, the genome is plus-stranded RNA. It has the same polarity like an mRNA, and in fact, it can be expressed as such, and then creates a negative sense anti-genome and negative sense anti-specific mRNAs to be able to really amplify expression of this specific RNA that codes for each of those proteins. I'll go specifically through how this is occurring. Again, the nidovirales, nidoviruses are really, the nido really depicts the nested loops of RNAs that are really created. And these nested loops means that all mRNAs that are created have the same 5' and 3' end. And I'll go more specifically to tell you how this occurs. As Elijah mentioned, one of the very important proteins of the virus is the spike glycoprotein. It has a receptor binding specificity. It is really the fusogen. It is the glycoprotein that opens up a pore between the viral envelope and the cellular membrane. And then by energy minimization, this pore opens up and the envelope fuses with the plasma membrane, depositing the nucleocapsid into the cytoplasm. The same exact phenomenon occurs in the endosome, where the viral envelope fuses with the endosomal membrane, and then the nucleocapsid enters the cytoplasm. We know that the spike glycoprotein is activated by a number of proteases, and these proteases are important in separating and activating the fusogenic portion, which is really on the S2 or carboxy terminal part of the glycoprotein. The receptor is really on the S1 or the amino terminal portion, as is depicted on the right. We know that deletions in the S1 can alter the tissue tropism and the pH conformational changes in S, as well neutralization by antibodies really depend on the overall structure of the spike glycoprotein, which is a major antigen and immunogen that could potentially protect against COVID-19. So we know that deletions, the spike protein can change virulence, and we have examples from that from animal viruses. For instance, in a transmissible gastroenteritis virus, which is enteric disease in piglets, a deletion of 225 amino acids in the spike or specific mutations cause the porcine coronavirus disease, which is a respiratory disease in pigs, and it also causes interstitial pneumonia. So you can see that deletions and arrangements of the spike protein in terms of this receptor binding and function in fusion can alter the tropism of the virus. So this is the mechanism of how the spike really causes fusion. There's initial docking of the spike onto receptors. There are co-receptors for COVID-19. We know that this PMRSS2 as a protease that may be a co-receptor also. Co-receptors would function to increase the receptor binding. There's a conformational change of the spike protein and ultimately exposure of a heptide repeat region, which is depicted here as an alpha helix in red. And this trimer has now a 6-bundle heptide repeat that creates the pore for which really mediates fusion. And this is shown here on this very recent paper, how the mechanism of receptor binding ultimately opens up the spike glycoprotein, and the fusion occurs by the heptide repeat, heptide repeat that opens up the pore. You can actually see that the S1 domain of the spike, as well as the receptor here, which will be ACE2, would be cleaved by surface proteases to really fully activate the ability of the virus to enter into cells. And for coronaviruses and receptors, as I alluded to earlier, we have in different genera, we have the aminopeptidase N for human coronavirus 229E, which is a human respiratory pathogen. While we have also for the new viruses, we have the ACE2 receptor. And for others, at the genera 2, we have the carcinoembryonic antigen M1 and acetyl nucleic acid as also being sialic acid as being a receptor. And if you look now at the spikes of different proteins, you could see that the different arrangement in different viruses include different amino termini that modulate receptor binding, as well as where the areas of where the receptor may be located for different human and animal coronaviruses. But basically, the properties of the virus retains its ability to bind to receptors and co-receptors activated by proteases and ultimately cause fusion. So let me see, here we are, yeah. So in the early 2000s, in 2005 and 2007, Chad Petit, the graduate student in my lab, worked extensively on understanding the structure and function of the spike glycoprotein. And these are the two papers that we published in 5 and 7. And we showed that through site-directed immunogenesis that the fusion peptide, the heptide repeat region, is extremely important for the fusiogenicity of the virus in transient transfection experiments. And for the first time, we showed that the carboxy terminus of this spike glycoprotein, which is really inside the cytoplasm that contains a cysteine-rich region, was also very important in modulating the fusiogenicity of the spike glycoprotein, indicating, as we've seen for herpes viruses, that the carboxy terminus of the glycoprotein, although it's really on the opposed membrane inside the cytoplasm, could really alter the conformation and fusiogenicity of the virus. And these are also, we already visited some of the structures with Elia, showing the S1 subunit and the S2 subunit, and underneath the alpha helical structures, as you can see here, and showing the different important areas where it's the binding domain, the transmembrane, and S1, S2 is the area where it's cleaved by different proteases, and there are probably more than one protease involved in processing this protein. And this is an alignment showing the different coronaviruses that has recently appeared in Nature Medicine. And one of the main big importances or major differences between the SARS and SARS-2 is the fact that the SARS-2 contains a polybasic region between S1 and S2 that could also be cleaved by furin protease. And there is a lot of debate whether that polybasic region may be contributing to the infection and pathogenicity of this particular virus. And what we did in our laboratory, and I think this is now what we like to disclose to you as our new findings, and they will be ahead of publication, they will be submitted to publication probably either tomorrow or Monday morning. This is the schematic structure that Dr. Farhana Musharat has created in our laboratory of the SARS-2 spike glycoprotein, showing the S1, S2 domain, the receptor binding domain in green, the cleavage S1, S2, as well as 2' cleavage processing between cleaving the molecule, as is shown here. So we ended up with Dr. Vladimir Chouljenko in the lab, cloned this spike glycoprotein from SARS-2 coronavirus in transient expression vectors, and we ended up comparing its ability to fuse cells in African green monkey cells between the old version and the new version. And we also created expression plasmids that contain the amino terminus and the carboxy terminus that you see in C and D here, separately expressed by transient expression, asking the question whether really the independent parts of spike, if they were transfected and expressed at the same time, could actually cause fusion. And what we saw, it's a very drastic difference between the ability of the old spike glycoprotein, is called here SO, depicting the SARS spike glycoprotein, and S new is the SARS-2 spike glycoprotein. And you can see the phase contrast pictures of African green monkey kidney cells and at the top, and then stained with anti-spike antibodies. Both of those proteins have epitope tags in as fusions at the amino termini of both spikes and can be readily detected. And as you can see, the SN at the lower central panel causes extensive fusion, practically fuses the entire dish into large syncytia, although the old spike causes much more limited fusion. And then when you be able to try to see whether you reconstitute fusion by expressing the S1 and S2 together, neither the SARS spike or the SARS-2 spike actually are able to reconstitute it, meaning that the molecule has to be intact and expressed on most likely on cell surfaces before activation can occur in the right way to be able to mediate cell fusion. We went further than that and asked the question, are there any drugs that are in the market that could inhibit spike-mediated fusion? And Farhana in the laboratory screened a number of very interesting compounds that are already in the market. We'll only discuss here one of those that is actually a current drug, which is on the market for treating against the HIV infection by inhibiting the protease encoded by the HIV virus. And this compound is Nelfinavir. Nelfinavir was created as a specific protease inhibitor for the HIV protease. It has also been shown to have some activity against the proteases for SARS, and it has been shown to have ability to inhibit replication, at least in SARS-2, in tissue cultures. But what we saw here, what Farhana showed, is that on the very top, you see the controls with the new SARS spike glycoprotein versus the old, extensive fusion on the spike new glycoprotein, SARS-2, versus the old. And then at the 10 micromolar range for Nelfinavir, you can see complete inhibition of spike-mediated fusion. This indicates that this protein, this particular compound, if it really acts specifically against spike glycoprotein, would limit spread of the virus from one cell to the other. And our hypothesis is that this syncytia formation and cell fusion is really the hallmark of how this virus is spread, escaping the outside humoral immune response and neutralizing antibody. We went further and asked our collaborator from the Louisiana Biomedical Research Network, Dr. Seetharama Jois, who is working on a project for LBRN, to look to see whether the Nelfinavir has a different target. Does it target spike specifically, or is it really doing something different? Is it possible that it really targets the spike, although it really was first created against proteases? Also, I would be reminiscent to also tell you that Nelfinavir has been also used extensively as an anti-cancer drug for leukemias and also different cancers. And it does have a pleiotropic effect on cells, including inhibiting AKT signaling, apoptosis, created increased apoptosis, and a number of other potential cellular related issues. But here, at this concentrations used, we saw no aberrant, no toxicity really related to any of these potential effects. So Dr. Seetharama Jois, with his graduate student, I cannot remember the name right now. Hopefully Seetharama would chat with me and tell me. Ended up doing
Achyut Dahal.
Okay, got it. And so Seetharama did docking experiments with Nelfinavir onto the spike protein and selected these regions for specific docking experiments to actually see if Nelfinavir would bind to the spike protein. And very surprisingly, and of course exciting to us, was the fact that he indicated that Nelfinavir may be binding directly to nearly the heptide repeat 1 and the fusion peptide, as it's shown here. So it may have a direct effect on spike-mediated fusion. And I think in further seminars later on, we'll elaborate on those. There are a lot of other experiments ongoing to actually see if it inhibits cleavage and so on. But at least at this point, our recommendation is that Nelfinavir is a potent inhibitor of spike-mediated fusion, and it will be most likely a very potent inhibitor of SARS-2 spread in different types of cells, and also will be testing in pneumocytes. But let me go back and revisit the molecular biology of the viruses, because we went into a lot of the spike structure and function to tell you some of our results. This is the genome of the mouse hepatitis virus. There are 30,000 nucleotides. Ideally, you would think that it will code maybe 20 to 30 genes, different proteins. In fact, this virus codes almost 50 different proteins. And it does that by creating, using the compactness of the genome through different molecular biological phenomena to be able to code more proteins that the genome could probably contain. And as you can see here, the color regions are different domains that are being cleaved by proteases or really coding regions, including the spike protein. And you can see that a number of subgenomic messages are created, as is shown at the RNA level. And all these subgenomic messages have the same 5' with the red dot and the same 3' with the poly A tail. So they're really both 5' and 3' identical. And if you look also at the entire genome and compare those viruses, you will see that those viruses have a similar arrangement of genes, but they also do have a number of other genes that are, some of them or most of them may be dispensable for viral replication in vitro, but they may be required in vivo, in particular for really defending against immune responses, down-regulating innate immune responses and downstream immune responses. And you can see that in terms of the SARS coronavirus and SARS-2 coronavirus, there are some minor differences in these accessory genes, and I will discuss some of those functions as we go forward. And you can see that the polyprotein that creates the RNA-dependent RNA polymerase has two operating frames, ORF1A and 1B, and in fact, it ended up using both of these frames to create the whole enzyme, which is the RNA-dependent RNA polymerase. So this is the coronavirus transcription and replication. This is a plasmid RNA genome. It creates a negative stranded full genome. That's the intermediate negative stranded genome. It uses that then to amplify and create the genome RNA from the intermediate negative stranded RNA. The mRNAs are also in the same way created by splicing the specific leader sequence from the 5' end through the ability of the RNA-dependent RNA polymerase to jump from, as it really transcribes the negative stranded, jump from one specific segment having a specific regulatory sequence to the leader sequence to create a subgenomic message that has the same 5' leader sequence. And this jumping occurs by having this the regulatory regions where basically you have the negative strand created, the polymerase jumps to the 5' end, and then creates the co-terminal 5' co-terminal messages. This is a way to amplify, create subgenomic negative stranded RNAs that then are used to really express large amounts of positive stranded RNAs, mRNAs that will express each protein. And this is shown again here, where you can see that the negative strand subgenomic message, genome creates the plus-stranded subgenomic mRNA. And then ultimately, we'll code, for instance, for example, the spike protein. And these are really all the intermediates. We have multiple subgenomic negative stranded RNAs that create the mRNAs for each and a higher level amplification. What is probably the reason for that? The reason is that it can create a pool of individual templates, negative stranded, that could be used to heavily express mRNAs for expression purposes. And again, for the 2019 coronavirus type 2, it possesses 14 operating frames, but it does encode 27 proteins. And it's shown underneath here. And some of the accessory genes, 7A, B, and P6, are known. Some of them we know their functions, some of them we do not. There's a lot of intense interest in understanding how these genes function, because certain deletions of these genes may be associated with lower virulence. Now, the virus uses programmed ribosomal shifting to be able to generate two polyproteins encoding the replicase protein. And as you saw earlier, we have ORF1A and ORF1B that codes the RNA-dependent polymerase. It turns out somewhere in the middle between ORF1A and 1B, there is a pseudonut and a slipping ribosomal slippery heptanucleotide nucleotide that you see here. And as the RNA polymerase is processing that message, it could actually slip back up by one trinucleotide and then read through to ORF1b, creating an ORF1a, 1b polyprotein. And that polyprotein then can be cleaved by a number of different proteases to create different parts of the RNA-dependent RNA polymerase. And this is shown here. This is now the coronavirus replicase that has the RNA polymerase. It has also parts of the protein that include for mRNA capping and also proofreading activity. So this is really a big enzyme that has these proteins. The whole enzyme is composed of NSP7, NSP8, and 12. And NSP14, the non-structural protein 14, is actually really involved in the whole enzyme. With these proteins creates the whole enzyme. And the exonuclease is really the NSP14. And mutagenesis of the NSP14, which is the exonuclease, that also is part of this what's called proofreading capacity of the virus. If you really mutate that, then the virus creates tons of mutations very quickly and it doesn't survive. So this exonuclease activity and proofreading capacity is very important for creating stable genomes. The virus replicates in the cytoplasm, and we call replication transcriptional complexes. It creates these double bilayer membranes where the nucleoprotein bound to the RNA creates the subgenomic messages, and beyond that, as you'll see here. And if you're by microscopy, you'll see these vacuoles within the cytoplasm of infected cells. They're really the RTCs, the replication transcriptional complexes of the virus, where the replication will occur. And then again, after the virus replicates with the RTCs, shown here on the left is the SARS versus MERS on the right. The virus then will, through a reverse exocytosis pathway, will be coming back to the surface and will be excreted as a fully enveloped virus in these extracellular spaces. Now, I wanted to briefly tell you about potential targets for SARS coronavirus therapy, and I added those slides and a little bit of the immunology, and we'll go a little bit over time, but I had scheduled this for about an hour and a half, so we still have about 20 minutes to be able to discuss this. So obviously, if you have major antigens expressed on cell surfaces, like the spike glycoprotein, that would be a candidate for a vaccine. We know that neutralizing antibodies against the spike protein can be created in human beings and can be neutralizing the virus. So an effective vaccine should create high levels of neutralizing antibody. Also, we do know that the spike protein binds to specific receptors like the angiotensin converting enzyme 2 receptor. So fusion inhibitors that either inhibit fusion or inhibit binding to the receptor would also be a way to combat this particular virus. In addition to that, we know that the virus enters into cell, and one of the first things that we'll discuss in a few slides further down is that the virus controls innate immune responses, and especially interferon, type 1 interferon responses. It has a number of different molecules that will downregulate the interferon response because it wants to replicate, and ultimately the exacerbated immune response is really what causes the pathogenesis and death, as it happens with other pathogens, as we've seen in sepsis with bacterial infections. In addition to that, the virus obviously transcribes, so inhibitors against the RNA polymerase, the enzymes, or inhibitors of the proteases. All of these could be tested, and in fact, a number of them are really now used. There is remdesivir that you've seen in the news that was created for a different virus, as well as Vladimir for flu, that has been used in Japan as a specific RNA polymerase inhibitor with some encouraging results in clinical studies. Now, as I told you, the coronavirus and SARS have a similar sets of accessory genes, slightly different, and some of them are the differences really are particularly important as interferon antagonists. And these are the accessory proteins on the left, and you can see whether they incorporate the virions or VLPs, and they have properties for NF-kappa beta activation, the type 1 interferon responses, and of course apoptosis induction, cellular DNA synthesis, and so on. So some of these proteins, especially the ones that relate to type 1 interferon, will be particularly important to really create potentially even live attenuated vaccines that could protect against the virus. Now, therapeutics that, as I said earlier, this is for a recent article that I should have the where it's from underneath, but it doesn't show for some reason, that you can see the different inhibitors. So phosphoprotease inhibitors have been shown to be effective in mouse models, monoclonal antibodies, interferons, ribavirin, and of course protease inhibitors that be used off-label for patients, including ribavirin right now. And then the hallmark of this infection is actually highlighted here in the whole cell host and microbe that appeared for mouse hepatitis virus. And the hallmark for the importance of interferon is shown on the upper left, where mice that were interferon receptor negative survived the SARS infection versus mice that were not. So interferon response was really important in terms of survivability. And at the D panel, you see that the viral replication in these mice were really the same. And the authors explained that what may be happening for SARS is that you have a delayed interferon response because of downregulation of interferon by the cellular, the viral genes that are embedded within the virus. And ultimately, you have a delayed response that leads to a dysregulated inflammatory monocyte macrophages response, and you create lung immunopathology and ultimately lethal pneumonia. And that's there is for the general pathology of 2015. The idea is that you have these pneumocytes that are infected, you have an acute infection, a number of inflammatory cytokines that are expressed, including TNF, IL-6, and IL-beta, and IL-1 beta, and CCL2. And ultimately, you're creating acute lung injury and acute respiratory disease because of this cytokine storm. And in this particular paper in 2019, that was specific for MERS, they showed and hypothesized that interaction of the virus creates a cellular immune differential response that ultimately affects TH1, TH17 cells, that has ultimately the infection is controlled by interferon gamma and granzyme, but in the process of this, you create an exacerbated immune response that causes really the immunopathogenesis of the infection and resultant death. And one more note on the neutralizing antibodies. We do know that from human patients, and this is shown from Journal of Immunology 2011, that for SARS patients, that the immune response, the neutralizing antibodies actually wane out over time. And you can see the average of the cohort shown in black, that in months after 20 to 30 months afterwards, the neutralizing antibody is gone. And one of the issues is whether a vaccination approach could create a memory B cell response that could really create neutralizing antibodies that would be long-term protecting. So in terms of coronavirus vaccines, we know that we have created vaccines for animals. We have, for instance, live attenuated vaccines, infectious bronchitis virus, this or the porcine enteric disease virus that passes freely in viral cells and ultimately become a virulent. We have kill vaccines that have been used, as well as vector vaccines. And of course, passive immunization that's already used in human patients, creating a serum transfers could be potentially used to treat human patients. But there's also a number of reports for SARS in non-human primates, as well as in mice, that a low antibody response could create antibody-dependent enhancement. It's also known for the feline coronavirus vaccines, which means that extreme care has to be taken that the immune response has a high level of neutralizing antibodies, because low levels of antibodies that just bind may actually enhance the ability of the virus to infect, meaning that people that have passed this virus would still be susceptible to be reinfected by the same virus. And these are vaccine strategies that have been used by a recent review article. And I want to elaborate on this because I already talked about this. And there's a number of different approaches that could be used for vaccine purposes. We are at 1:20. I wanted to leave a little bit of time for potential questions. For those, I want to make sure that I acknowledge the people that contributed to this presentation, in addition to Elia, who will be working together on a number of different fronts for research, as well as developing courses and training modules for bioinformatics throughout the LBRN network. Farhana Musarat has done all the transient transfections that you've seen, and has been working with me for publishing this paper. And of course, Vladimir Chouljenko is a resident expert molecular virologist that has cloned all these genes. He's actually in the process of sequencing some Louisiana isolates through our gene lab core, which is supported by the LBRN, and also the lung COBRI, which is headed by Drs. Seetharama Jois. And Muzammel Haq is already involved with us in expressing these proteins for potential testing these antigens for the presence of antibodies in human patients and perhaps also in animals. I want to thank you for attending. This was an impromptu seminar to bring everybody up to speed on the molecular biology and hints of immunopathogenesis of the virus. And we could take some questions at this point.
Hi, I have one question.
Yeah.
Among the proteins involved in COVID virus, I think replicase is one of the most unique enzyme which it does not have homologue in the humans. Then why that shouldn't be a primary target of inhibition?
Yeah, so really, you're absolutely right. And a lot of people now, I'm sure the three-dimensional structure of the enzyme itself has been solved. So a lot of people doing in silico docking experiments to see if they can actually design specific inhibitors. But right now they're using mostly RNA pole active site inhibitors from other viruses, including Ebola and influenza. And those potentially bind, but most likely they'll not bind to the high affinity that may be required to really inhibit that enzyme.
Yeah, thank you.
Yeah, hi, Gus. I have a question.
Yes.
Elia.
Yeah.
This is Paul.
Sure. Yeah, hey, Paul.
Hi, this is for Elia. So you showed that there's 25 genomes per week, or was it per month, for this virus that are being sequenced?
Yes, this is on average per month.
Okay. Have you noticed any differences in the mutations as it spreads through the human population?
Yeah, so I mean, first of all, this is already documented on GIS-8 that there are several different substrains that could be detected, at least 3, some propose 5. I think, that's based on the full genome. We specifically looked at mutations that are emerging, comparing different groups from different locations. For example, we have some comparisons between US and Australian, as well as Chinese patients. And there are some, they're infrequent. So in other words, it's difficult to estimate how representative this is of the wider population, just because the sample size is not that big. And we are looking here for whole genome sequencing, raw data. But yes, some interesting mutations have been emerging. And of course, that has significance for vaccine design and things like that.
All right, interesting.
Hey, this is Ramesh. there is really a report that there is it's a strain in India which has mutation in the spike protein or rendering unable to bind to the ACE2, Mr. Do you comment on that?
Is that from me or from Gus?
What did you say, Ramesh? I didn't hear your question. Go ahead, because you were breaking up a little bit.
So there was this report from that said that there's a mutation in spike protein which renders it unable to bind to the ACE receptor. I was wondering if you have any comment on that.
Yeah, I don't know. Is that a recent? There was a couple genotypes that showed that as the virus evolves, it may be losing its affinity, the high affinity for the ACE2 receptor. That's all very recent. I'm not sure whether it's correct or not.
And I think that in that same report that you're proposing, that you were referring to, I mean, you know, there's a question of how many and how, you know, accurate the sequencing quality is and things like that. But what they were saying in that publication is that it reduces the binding of the receptor binding domain with the ACE2. And, you know, that should be a good thing, I think, right? From the standpoint of that publication, yeah. I think they were proposing it's maybe.
So what may be happening is, and I think that's the expectation, that as the virus really propagates in human beings, in an anthropomorphic way, the virus really doesn't want to really kill the host. So ultimately, you're going to have a minor deletion somewhere, one of the accessory proteins, or you may have an alteration in the spike protein, and the virus will be able to come into equilibrium and ultimately will become, hopefully, one more of the human coronaviruses that cause fairly mild respiratory disease. Now, when that equilibrium will occur and how long will it take and will it be that way, that's really subject to debate.
Hi, Gus. This is Weishan. I already typed my questions in the chatting window.
Yeah.
But just in case you haven't seen it yet.
I haven't seen it. Go ahead, Weishan.
Yes. My first question is about the blocking of the cell-cell fusion, trying to screen for candidate drug. So the Nelfinavir, is it the right pronunciation?
Nelfinavir, yeah.
Yes. So do you think it's blocking the binding and entry of the virus into the cell so that it blocks the downstream cell-cell fusion, or is actually targeting more downstream events?
So basically, our hypothesis is that this virus really spreads from cell to cell, primarily through fusion, that most likely not that much of free virus is really deposited in lungs. So the ability of the virus to really spread from cell to cell and create large syncytia is also explains why you see these very focal areas in lung x-rays that spread in a localized way. Because if you had a disseminated viral infection, you would see infection throughout the lung. So at least it's just a hypothesis that the ability of the virus, and remember, the spike-mediated cell fusion is really a surrogate of membrane fusion, even for virus entry, because it does require the ACE2 receptor and appropriate processing. So we do not know whether this particular drug, Nelfinavir, at this point, alters the protease and different protease, because as you see in these transient transfections, there is no protease encoded by the virus. If there is an outside chance that this particular drug may inhibit a cellular protease, but that we will know fairly quickly, probably the next few days, if that's the case. If it does inhibit directly by binding to have the repeat, as well as Dr. Jois has proposed, then it is very possible that would inhibit during entry. That is, if the drug is in solution, and when the molecule is cleaved to activate, if the drug binds to the heptide repeat and the fusion peptide, the prediction is you would inhibit even virus entry.
So I was trying to think of a technical method. So if we want to segregate the two events and study specifically the upstream binding entry and the downstream cell-cell fusion, how could we design the experiment?
Yeah, obviously, you know, we're collaborating with you, and I think we're going to be going after VSV pseudotype variants, which is one way to test whether spike protein can function during entry. The ideal experiment really is to test it against the real virus, the SARS-2 virus, and that could be done by having the Nelfinavir not added to cells, but really added at the time of infection, because then you would eliminate the possibility that there is a cellular effect on the drug. You could also pre-treat the virus, although you would expect that the Nelfinavir wouldn't bind to the closed spike protein. Nelfinavir will only bind if the spike protein is proteolytically activated, allowing the heptide repeats to be exposed. So basically, it would only function during a very short period when the spike glycoprotein mediates membrane fusion.
Thank you.
Dr. Kousoulas.
Yes.
There are some questions in the chat box after Dr. Vishen's question.
Yeah, can you, because I don't see my chat box here, can you read one of those?
So the second question is, would antibody-dependent enhancement of viral replication manifest as worsening disease?
So, you know, the phenomenon of antibody-dependent enhancement is known for other viruses. For instance, dengue viruses, that infection of one serotype makes you more susceptible for a secondary infection by a different serotype. It also has been documented with SARS in non-human primates in one experiment where basically 3 monkeys were vaccinated with inactivated virus, and then they were able to challenge them. One of the monkeys came down with acute lethal pneumonia and died. In addition to that, certain mice experiments have shown with SARS that antibody-dependent enhancement is very possible. So I think creating the right type of immune response, long-term, high-level neutralizing antibodies, and with avoiding potentially antibody-dependent enhancement would be very important, and it would be a difficult issue as people progress into vaccine strategies.
Okay, so the following question is there any idea what makes this COVID-19 highly infectious?
So basically, what people have found is that the COVID-19 spike glycoprotein binds to the ACE2 receptor at approximately 20 times stronger than SARS. So the binding to the ACE2 receptor is much stronger, which maybe then increases the infectivity of the virus, and also reduces the potential viral load required for infection. The better the attachment, the better the entry, the lower the infectious dose for infection, and that's why perhaps this virus has such a high transmittability and ability to be transmitted.
Okay, so there's another question from Dr. Vishen. Do you have any comment on the glycosylation that can affect the immunogenicity of S protein?
Yes, and I think Elia has done some wonderful comparisons of where the glycosylation sites, the spike glycoprotein is both N-glycosylated on asparagine as well as O-glycosylated. There are substantial differences in glycosylation between SARS and SARS-2 spike glycoproteins. Those carbohydrates could be used to shield potential neutralizing epitopes, or alter the conformation of the protein itself. So a systematic mutagenesis of these glycoproteins will be needed to really address whether some of these differences are really alter the functional properties of the spike protein, as well also its ability to be neutralized by neutralizing antibodies.
Okay. So there's one more question on the infectious and virulence. I think you already answered that. So there's a general question. What's your opinion on the lockdown strategy?
So we know that social distancing is the most effective way of really lowering transmission. And the social distancing is really the most effective strategy to protect the health of workers and ability of hospitals to be able to deal with the infection. In reality, if we had adequate hospitals and adequate ability to really deal with thousands of these patients, perhaps the social distancing may not be that important, but the social distancing is really to slow down, flatten the curve, so you can actually able to deal with it with regard to healthcare. There are a number of countries that moved very quickly to we have a number of different rules for social distancing. I know that, for instance, in Europe, Greece has only 100 deaths in a population of nearly 12 million, with only a few thousand cases. But they really, because they were scared because of the Italian epidemic up in North Italy, they ended up putting heavy fines and really slowing down the infection rate. And so far they're doing great. The Wuhan example is also very important. You only have under 4,000 deaths. The Chinese government acted very quickly to really slow down everything. Unfortunately for us, we do not see that here in the States. We have inability to probably work together across all states. There are states that compete with other states for protective equipment and other things, and there are different rules or regulations everywhere. This is part of the problem we're having in democracy, and very hard to convince people to really stay home and really exercise effective social distancing.
Thank you. So we do have some international listeners. Dr. Shangita Ahmed, she's from Bangladesh. So her question is, as far as we know, there are 3 types of viruses globally circulating. Do you have any data on the virulence of those different strains?
Well, early publications showed at least 2 genotypes, more virulent and less virulent. The more virulent appeared in North Italy. It's really very interesting in Italy what has been happening, because there is high degree of death rate and infection rate in northern Italy, but not in the south. For instance, the city of Ferrara has practically no infections. And there's also an issue there whether there is really different virus strains, or is it really some kind of a genotype, a human genetic variation that may cause resistance to this infection. And I think it's too early to tell. There's been recent publications, they've been identified more than 12 types of coronaviruses circulating. Whether any of these viruses are really linked to substantially more virulence is, I think, subject to debate.
So the next question is from Tim. I think it's about viral fitness. So what Tim is saying, doesn't the selective advantage of the high affinity binding site imply that mutants with lower affinity will be outcompeted, thus maintaining high virulence?
Yeah, that's actually a very interesting question, because obviously virulence is not just a factor of the spike. I'm sure the RNA-dependent polymerase, the accessory proteins, the interferon response, all of these are really part of the equation. looking at the molecular evolution of the virus. So when I mentioned ultimately that the virus may become less virulent, it would be the composite picture of the genomic variation to adapt the virus to live in harmony with the host. But having a stronger binding versus lower binding and how this would affect the fitness of the virus, one would argue that the lesser affinity, the more the ability of the virus to survive within the host and cause milder disease. Then again, in an anthropomorphic way, I don't think the virus really wants to kill human patients. Part of this coevolution of these viruses with human beings is ultimately to come at some equilibrium and cause a milder disease that could be dealt with by human beings. Unfortunately, most of us do not evolve as quickly as those viruses.
Okay, thank you. So the next question is from Dr. Mahbub Hossain from Bangladesh. So he's asking, can hydroxychloroquine, or is there any possibility that it will prevent the infection? Can it work prophylactically?
So hydroxychloroquine is actually a drug that is found to be is being utilized in all trials. The recent clinical trials show that it has not been effective for people that have acute pneumonia that are intubated in hospitals. It's also interesting that this particular drug is used extensively at low doses for people that have lupus or autoimmune responses. And it does really act to alter the cytoskeletal network of cells. And it is really, at least in tissue culture, has some effectiveness against viral infection. So is it possible that low doses of hydroxychloroquine could be used on a potential preventive way? It's already pretty safe if it's used under, I think, 500 milligrams per day. But in those experiments, they tried more than a gram, 1.5 grams per day. And I would think, from what I read, that would cause more problems than actually help.
Thank you. So there are two more questions from Lindia. What the question is, could antibody-dependent enhancement theoretically can lead to globally higher infection rates in convalescent individual later on?
Well, this is a hypothetical argument. Obviously, what we know from dengue viruses, it is possible that you could have local epidemics that depend on antibody-dependent enhancement. Ultimately, that could be also needed, containment would be needed for that. To just like maybe the comparison to earthquakes, you have a major earthquake and then you have a number of tremors afterwards. I think in a comparative way, I think we will see not only this pandemic, but variations of this as local epidemics probably the next 5 years.
Okay, thank you. Question from Dr. Plata. Is there any data that how much cross protection can be observed or exists between SARS-CoV-1 and CoV-2?
Yeah, that's a very interesting question, and I've done a lot of literature searches to find some of that. It hasn't appeared yet in the literature. I don't know if Washan that's listening may have some comments on that. But if you look at the repertoire of BNT cell epitopes, there are certain epitopes that are shared. So I think it's possible that some level of cross protection will actually exist.
Is there any other question?
I have one last question.
Yes.
Yeah. Is this spike-mediated viral fusion to host is unique to this COVID-19, or that is shared between other COVID viruses?
Yeah, so all coronaviruses, whether animal or human coronaviruses, use spike to enter into cells and spread. Interestingly, now some coronaviruses do not necessarily cleave S1, S2 and still mediate fusion. There are other fusogenic glycoproteins, for instance, herpes glycoprotein B, that's not cleaved, that's not really required to be cleaved. to expose the fusion peptide. Obviously, if you cleave and keep the heterodimer together, you seem to be much more active in fusion. But the answer to your question is that all coronaviridae do use spike as the main mechanism of entry and spread.
The reason why I asked the question is, well, unless I'm wrong, but hemagglutinin protein is mostly involved in fusion events, and why this has additional mechanism for fusions.
Yeah, so hemagglutinin esterase is actually in some of the animal viruses, and it's also used to really facilitate entry, but the actual fusion is actually caused by the spike glycoprotein.
Okay, thanks.
Anyway, we're way over time by 15 minutes. It was targeted to 1:30. I want to thank everybody for listening. We had over 100 people at the peak time. Thank you for listening, and we'd love to hear from you. Any comments that you may have or any questions that you may have, we will try to answer them. Obviously, Ilya Brodsky has a complete course on using bioinformatics to address important issues in molecular virology. We'll be working with him to sponsor this particular series of seminars, and we'll have additional seminars hopefully in the future that address specific aspects of coronavirology and immunopathogenesis. Thank you for attending, and be safe.